Just before that, some functions of CSF. It can come as a short type question. First is the mechanical support of brain and spinal cord. Second is the in maintenance of intracranial pressure. Third is chemical buffering. Fourth is transport of neurochemical substances, including growth factors. So these are the basic functions of the CSF. First is the mechanical support of brain and spinal cord. Since the CSF is present at the subarachnoid space, and you know that once a fluid is present, it provides some sort of buoyancy. So it makes the brain relatively lighter. Buoyancy તમે જાણીચો? Whenever a ship is floating on water, the water also exerts some force on the ship to keep it afloat. So in the process, it makes the weight of the ship a bit lighter. So in that case, since CSF flows in the subarachnoid space, that makes the entire brain structures relatively lighter and provides that way it provides a support to the brain and spinal cord. Secondly, maintenance of intracranial pressure. So all the means suppose there is a change in the blood pressure, immediately the pressure in the CSF that is not being changed, and in this way the pressure is maintained a bit, or the fluctuation of the pressure is adjusted a bit. So inside the cranial means in the lateral ventricles, in the third and the fourth ventricles, it helps in the maintenance of the pressure, or there's an intracranial pressure whose maintenance is also a function of this CSF. Thirdly, chemical buffering. We said that certain waste products of the CNS are also are also excreted into the CSF, and in this way CSF also helps in chemical buffering. And fourthly, transport of neurochemical substances, including growth factors. That is also one of the functions of CSF. Then coming to the site of collection, sometimes CSF is collected to detect pathology of the CNS. For examples, presence of RBC, hemoglobin breakdown products, inflammatory cells, elevated proteins. They can be collected from the cisterna magna. That is a point which is located caudal to the cerebellum, and from subarachnoid space in the lumbar section of the spinal cord. So these are the two sites from where CSF can be collected, so that you can examine them for any pathological problems, like there is any presence of RBCs or hemoglobin breakdown products or inflammatory cells, or if the proteins are elevated, that you can detect, and you can say that yes, there is a problem with the brain, or there is a problem with the nervous system, or there is any discrepancies or any abnormalities in the CSF or in the constituent of CSF. Now you can see from the caudal portion of the cerebellum that there is a point which is known as the cisterna magna, from which you can see there is a pipe which is used to collect the CSF. Then also in the lumbar section of the spinal cord, in the subarachnoid space, you can see the CSF is being collected from this point. Then coming to blood-brain barrier. So in the left panel, you can see a general capillary of the body. In the right panel, you can see the capillaries of the brain. So blood-brain barrier, that means just like blood CSF barrier, between blood and brain, there must be certain barriers, so that whatever substances of the blood, they are not able to reach the brain, because brain is highly sensitive. The brain components, they have got different sort of receptors also. Any exposure to any sort of toxins or higher concentration of something, that is also going to destabilize them. So all the things of the blood should not reach the brain. That is why we have got a blood-brain barrier. And in the left panel, you can see a general capillary. You can see these are the endothelial cells, and in between the endothelial cells, we have got certain intercellular clefts. This is the basement membrane, and also we can have certain fenestrations. As we can see, as we saw in case of capillaries of the kidneys or the glomerular capillaries. In case of this choroid plexus, we had got fenestrations. So this is the structure of a general capillaries. But what are the different barrier components, or what are the different components of the capillaries of the brain, and how it looks like? Here you can see between the endothelial cells, we have got tight junctions. Tight junctions are present between the endothelial cells. Apart from that, you can see, you remember the neuroglial cells, astrocytes. These astrocytes were star-shaped cells. They used to extend their processes, and the terminal end of the processes were called as perivascular end feet. We had discussed in the previous classes. So these perivascular end feet processes of the astrocytes, they usually cover the capillaries of the brain. You can see four different astrocytes, or you can see the astrocytic processes from four different directions. They are covering the capillaries of the brain. So the brain capillaries, these tight junctions will form a barrier function. These astrocyte perivascular end feet will also form a barrier function. That is why if you are asked which cell is responsible for formation of the blood-brain barrier, your answer will be astrocytes, or the perivascular end feet or end processes of the astrocytes. So you can see the capillaries, the general capillaries of the body, they do not have certain barrier functions, but the capillaries of the brain, their arrangement of the endothelial cell is different. You can see between the endothelial cells there are tight junctions, and there are also certain perivascular end feet which are covering the capillaries. So this is how a brain capillary is different from general capillaries. That is why everything from the blood will not reach into the brain. Now, capillaries of the CNS, they are associated with three unique features as we discussed right now. First is continuous tight junctions that seal neighboring endothelial cells. Second is there is absence of fenestrations, because we are saying about the capillaries of CNS. Previously, we were saying about the capillaries of the choroid plexus only. So in the capillaries of the CNS, there is absence of fenestrations. And thirdly, only a small number of pinocytic vesicles. So these are the three characteristic features or the unique features of the capillaries of the CNS. So although capillary endothelium is the structural basis of the blood-brain barrier, such a property appears to be maintained by astrocytes that form perivascular end feet around the entire outer surface of the capillary endothelium. Got it? Now, if you are providing this blood-brain barrier to each and every area of the brain, then certain functions can be modified, or certain functions cannot be achieved. How? For example, if you provide blood-brain barrier to the choroid plexus, then your CSF can't be formed, because for formation of CSF you need fenestrations in the capillaries. So that the choroid plexus, that is devoid of blood-brain barrier. Secondly, pituitary or hypophysis. You know that pituitary, it is under the control of the hypothalamus. The hypothalamus releases certain releasing hormones which stimulate the pituitary gland to stimulate something. So if you are stimulating or if you are providing this blood-brain barrier to the pituitary, then also some functions will not be happening or will be affected. Thirdly, median eminence. Median eminence is a portion which is lower or towards the lower end of the hypothalamus. At those portions, some amount of hormones are also released. So if you provide blood-brain barrier to that portion, then these hormones cannot come to the level of pituitary. So these median eminence, that is also devoid of this blood-brain barrier. Fourthly, pineal gland. And finally, the area postrema. Area postrema is something that is present at the level of medulla oblongata, and its function, its job is to help in the process of vomission. Now, to initiate vomission, your vomission center should realize that whatever is present in the blood, that is toxic to the body. So everything that comes into the blood should reach the vomission center so that it can initiate vomission. But in that portion, in the area postrema, if you are providing blood-brain barrier, then the toxic substances or certain substances will not be able to reach those areas. So your vomission will not be initiated. And vomission is a very defensive mechanism also. Suppose you ingest some sort of toxins, you will vomit. Some sort of things which is affecting your osmolality, you will start vomiting. So this vomission center must not be a part of the, or it should not be provided with capillaries containing blood-brain barrier. So these are the non-barrier regions which include choroid plexus, the hypophysis, the median eminence, the pineal gland, and the area postrema. And capillaries in these areas are fenestrated, which is essential for these areas to carry out their functions. For example, release of hormones into circulation. For that, you need fenestrated capillaries. Monitoring circulating molecules, like area postrema is monitoring the molecules which are being circulated, so that if any sort of wrong substances or any sort of poisonous toxic substances comes, it will start to initiate vomission. So that portion we can't afford to supply them with blood-brain barrier. So these are the substances which are not having any sort of blood-brain barrier. Now we said that cerebrospinal fluid, that is 99% water, that also contains some amount of micronutrients like vitamin B6, but it can't supply the brain with glucose, amino acids, or the major nutrients that are being required by the brain. So that will be supplied by the endothelial cells or by the capillaries which are supplying the brain. So here you can see, this is the capillary lumen, this is the nervous tissue, and this is the luminal side which is exposed towards the lumen. This is the abluminal side, that means towards the nervous tissue. Now glucose, for you know that nervous tissue that utilizes glucose very much. You feel तमे जदि कंटिन्यूअसली बसिकी कंसंट्रेटेड वे रे घंटे रु 2 घंटा पढ़िथिबो, जदि पढ़िबो बसिकरी, you will start feeling hungry, because glucose is highly utilized by the brain during any sort of thing. So inside the nervous tissue, since glucose is utilized, and glucose utilization by the nervous tissue also is non-insulin dependent. That means nervous tissue, they do not require insulin, whose job is to help in the glucose transport into the cells or utilization of glucose by the cells. But nervous tissue, even if you are making them devoid of insulin, they can also help or they can also utilize glucose. So nervous tissue is one of the tissues which do not require the help of insulin for glucose utilization. So there is always a deficit of glucose inside the nervous tissue, because glucose is rapidly being consumed by the nervous tissues. That is why there are certain transporters which are known as the GLUT, right? And these transporter, they are present, suppose this GLUT1 is present at this point, which are helping in the transport of glucose from the blood into the nervous tissue by the process of facilitated diffusion. Why facilitated diffusion? Because from an area of higher concentration, glucose is moving towards an area of lower concentration. And nervous tissue, you know, that utilizes glucose to a prime extent. That is why the concentration of glucose is always in a deficit position than that of blood. So glucose will try to enter inside through this GLUT, which are the glucose transporters. Short GLUT, and the GLUT one is present here, whose job is to bring glucose inside. Secondly, we have the sodium potassium ATPase pump, sodium outside, potassium inside. So deficit sodium there is present, and sodium can also move through this. Then water can also move, and certain amino acids like leucine, they can, if necessary, they can move out from nervous tissue towards the blood. If they are required, they can also move from the blood towards the nervous tissue. Similarly, glycine, which is generally produced by the nervous tissue, they must be excreted out from the CNS towards the blood. So glycine takes the help of sodium symport to come inside. So based on the different requirement and the different amino acids, certain substances they can move towards the nervous tissue. Certain substances they can move both ways, like the amino acid leucine. Certain substances or certain amino acids which are produced by the brain or by the CNS, they must be taken out of the nervous tissue into the blood. So in details, if you see, the lipophilic substances like nicotine and ethanol, they are very permeable, and their transport through endothelial cells is only limited by blood flow. So lipophilic substances can easily cross from the blood into the brain. Similarly, gases like carbon dioxide, oxygen, nitrogen, they can diffuse rapidly into the brain. Water also crosses freely in either direction through the membrane by diffusion as the osmolality of the plasma changes. However, water-soluble substances, they are restricted from passing through the blood-brain barrier into the brain. And this glucose, which is a water-soluble substance and is a vital source of energy in the brain, and its transport depends on a specific glucose carrier, that is GLUT1, in the capillary endothelial cells. And GLUT1 is a facilitative transporter located at both the luminal and the abluminal side of the endothelial membrane. So you can see in the last diagram we showed how GLUT1 is present towards the luminal side, also at the abluminal side. This blue color carrier protein, which is the GLUT1, that is present at the luminal side and the abluminal side. So water can come inside. Any sort of gases like carbon dioxide, oxygen, nitrogen, they can also diffuse as such. Lipid soluble substances, since in the first class I had told you that the plasma membrane is composed of this bilayer. This is the cell membrane. You can see the hydrophilic layer here. We have the hydrophobic layer in the middle. Then we have also got the hydrophilic layer here. So they can, so any sort of lipid soluble substances, they can easily come from outside to inside. Right. Water can also move through the watery channels or through the pores. But any sort of lipid soluble, any sort of water soluble substances like glucose, amino acids, they need certain carrier proteins to come inside. And in this case, the carrier protein is the GLUT1, which is present both at the luminal side and the abluminal side, which is responsible for glucose transport inside the cell, or glucose transport from the blood into the nervous tissue. So the transcellular transport is the only way for any substance in the blood to enter into the CNS. The plasma membrane is made up of lipid bilayer. It is not permeable to charged particles and most polar molecules such as sugar and amino acids. Anions in water are attracted, leave it, not required. Then large neutral amino acids like phenylalanine, leucine, tyrosine, isoleucine, valine, tryptophan, methionine, histidine, and 1-DOPA, they are transported by facilitated diffusion both on the luminal and abluminal sides of the endothelial cells. Some of them, for example, tryptophan, which are the precursors of the neurotransmitters, serotonin and melatonin, they are synthesized in the CNS. Serotonin is involved in the mood and sleep, and melatonin regulates the sleep-wake cycle. Right? Similarly, smaller neutral amino acids such as glycine, alanine, serine, cysteine, proline, and GABA, they are synthesized in the CNS. So these amino acids, they are also transported primarily from the brain to the circulation. So large amino acids, they can move either ways, and small amino acids which are normally synthesized in the brain or in the CNS, they also can move outside from the CNS towards the blood. It appears that essential amino acids which are precursors for catecholamines like epinephrine or norepinephrine synthesized from tyrosine, and indolamine, which is example are serotonin and melatonin synthesized from tryptophan, they are transported into the CNS. Tyrosine CNS ଭିତରକୁ ଗଲେ ହିଁ तमर epinephrine ଆଉ norepinephrine synthesize ହବ, because these epinephrine norepinephrine they are dependent on the amino acid tyrosine. Tyrosine will be converted to dopa will be converted to dopamine, then that dopamine will enter into the vesicles inside which the norepinephrine and norepinephrine will be synthesized, जोटा कि मैं आगु को पढ़िबा. So जो substances गुडा CNS भितर को जिबा दरकार, so that neurotransmitter can be produced, those substances can be moved from the blood into the CNS. On the other hand, amino acids जोड़ा कि synthesize हला CNS रे and which function as neurotransmitters are not just restricted from crossing the blood-brain barrier into the CNS, but are transported out of the CNS. So substances, if required, they can be synthesized or they can be transported from blood into the CNS, and substances which are produced in the CNS, they can be transported from the CNS outside into the blood. Now this is a case of regeneration of nerve fiber. I had told you that if the cell body is destroyed, then there is no chance of regeneration of a nerve fiber. But if the axon is destroyed, suppose here you are cutting the axon, in the second phase what happens? A nerve sprout will be formed. Then these portions, the portions which are already destroyed, they will be degenerated by traumatic degeneration and Wallerian degeneration. And secondly, there will be thirdly, there will be production of a growth cone, and this is how the entire axon will be regenerated. And the Schwann cells, they will function in the process, and astrocytes also play a vitally important role in regeneration of nerve fibers. But thing is that if your cell body is destroyed, then there is no chance a neuron can be regenerated, because you know that the neurons, they stop dividing, but the glial cells, they can divide. So even if you are cutting an axon, there is chances of regeneration. Spinal cord injury hele, sort of, if somebody meets with an accident and the spinal cord is injured, in that case it takes a lot of time for the substance or the functioning of the body to come back, because there is continuous production of this nerve sprout, there is continuous production of this growth cone, the axons are regenerated, everything are regenerated. But if the cell body is damaged, then the function will be completely stopped, or there will be complete stoppage of the function. Now this is one difference between reflex, that is monosynaptic versus polysynaptic reflex. In this is, here you can see, this one is a knee hammer. I hope all of you can see. This is a knee hammer. Its job is to, if you go to a doctor and he wants to verify your reflexes, whether your reflex is functioning properly or not. So what he will do, he will take this knee hammer and start stroking your knees, or just the portion below your knees. So if the particular point is stroked, you will start moving your legs automatically. That is the knee jerk reflex we say. So doctor, what he does, he strokes your knees and you jerk your foot. And how this happens? By the help of a monosynaptic reflex. You can see if you are hitting this portion, then there are certain sensory receptors at this point which are taking the reflex through this pseudo unipolar neuron through the dorsal horn of the spinal cord into your dorsal root of the spinal cord, and it is coming like this, and at the ventral horn of the spinal cord it is contacting with a motor neuron, and this motor neuron, it is going and it is stimulating your effector organ, and you are able to move your feet. So this is a knee jerk reflex. It is a type of monosynaptic reflex because there is only one synapse involved. This is one sensory neuron. This is one motor neuron. There is only one synapse that is involved. But polysynaptic reflex, suppose you are touching a hot object, you immediately withdraw your hand. That is the case of a polysynaptic reflex. You can see the blue lines, they are coming like this. These are the sensory neurons. This is a pseudo unipolar neuron present at the level of dorsal root ganglion. Then they are moving through the dorsal root of the spinal cord. Then they are contacting small neurons which are known as the interneurons. Here you can see cell body, you can see the axon terminals. So they are contacting here, and these interneurons, they are contacting the motor neuron, and this motor neuron is helping you to remove your hand. So this is the difference between polysynaptic and monosynaptic, because here one synapse is involved. Here you can see here one synapse, here one synapse. This is this withdrawal reflex, that is an example of a polysynaptic reflex.
So,
And in yes,
so what is the necessary of here to add that interneuron? Without that also it could have the same results.
what is the necessity for adding this
Yes, so that
interneuron?
interneuron, yes, what does it does something different from that monosynaptic uh, reflex?
Okay, the difference is that monosynaptic reflex is a bit much faster than the polysynaptic reflex. Like we discussed about the different nerve fibers, A alpha, A beta, A gamma, A delta. So everything could have been achieved at 120 meter per second. Then why God has given that your pain reflexes will move in type C? Your temperature, touch, mechanoreceptors, they should move in type A. Why has nature built you in such a manner? Everything could have been possible through type A. Getting my point? So in this case, by the end of this, you will come to know that this monosynaptic is faster than the polysynaptic. Why it should be faster? Because this knee jerk reflex, suppose you are running, you are stepping on an imbalanced manner on certain things or certain grounds. Your reflex should function immediately. ହାତଟା ଆଇରନରେ ମାରିଛ, ଟିକେ ଡେରି ହେଇଗଲେ ମ୍ୟାକ୍ସିମମ୍ ରୁ ମ୍ୟାକ୍ସିମମ୍ କ'ଣ ହବ, ତମ ହାତର ଏଇ ପୋର୍ସନଟା ଜଳିଯିବ, ଫଟକା ହେଇଯିବ। ବଟ୍ ତମ ନି ଜର୍କ ରିଫ୍ଲେକ୍ସଟା ଯଦି ସ୍ଲୋ ହେଇଗଲା, ତାହେଲେ ତମର ଏଣ୍ଟାୟାର ବଡିଟା ତଳେ ପଡ଼ିଯିବାର ଚାନ୍ସେସ୍ ଅଛି। That is more dangerous. That is why the system is designed in such a manner that your monosynaptic reflex is provided to those points where a fast reflex is necessary. Your polysynaptic is provided to those points where a slow reflex is necessary. And there you can debate why not all the synapses are electrical synapse. What is the necessity of chemical synapse? All synapses could have been electrical. So this is the beauty of nature, you can say. They have provided certain things in such a manner that is very apt for the situation that is being provided. You need a very fast reflex so that whenever you are going, you are running, you are stepping on something, your knee is able to tell you that yes, this is the thing, so that you can either withdraw your feet or you can send the other feet or send signal to the other feet to counterbalance. Suppose tame gote godo uthei dela ara gote jemti thibo taku counterbalance kariya pai. So that is the necessity for this reflex, and when there is a provision and
सर
to
this
make
data
more
को
faster.
फास्ट नहीं करते तो data को फास्ट नहीं करते ताले एमसी का लो कि सिस्टम ओवरलोड है क्यों data को फास्ट नहीं लाओ बोले चलो सर
देखा दीटा का फास्ट ହେଇଥିଲେ ତମର ଯଦି what I can say, if your there is a portion of IQ ଜାଣିଛ କଣ? Intelligent quotient. Intelligent quotient is also regarded with some scales. ଜାଣିଛ କି ନାହିଁ?
yes sir
ହଁ, IQ ର highest scale ଟା କେତେ? Somebody, I don't remember it. I want you people to say me. There is a highest limit of IQ.
so 140 or 150
Any one of you who can say, who can also say yes with any one of you who agrees with
So
Suraj?
200. So 200.
Asutosh is saying 200. Anybody who agrees with Asutosh? Anybody? Anyone? So ଏଇଠି ମୁଁ ତମକୁ କହିବି कि 200 ताले मैक्सिमम कहीं ହେଲା, 400 तो ହେଇଥିଲା ଆହୁରି ବଢିଆ ହେଇଥାନ୍ତା. ଆମେ କହିଲେ saltatory conduction. We said about saltatory conduction. So why some fibers are there which are unmyelinated? Everything could have been myelinated. Yes or no? So with faster effects, faster thing, you need also faster energies. गोटे सिनाप्स है आरा सिनाप्स होची, सिनाप्टिक ट्रांसमिशन रे भी तमरा एनर्जी यूज होची, कैल्शियम यूज होची, which is also liberating or exocytosing the neurotransmitters, जोटा कि हम आगु को पढ़िबा. बुझि पालो कि नहीं? तो सही केस रे तम जदी सब जिंस को फास्ट करिया चेष्टा करिवो, then obviously the system can get also overloaded. So I'm not saying that this is a slow means vitally slow, because you know that speed at which you are able to withdraw your hand whenever you touch this thing. Getting me? But
yes sir
a more faster speed is required in case of a monosynaptic or in case of a knee jerk reflex, because there you are at more risk. एठी खाली हातटा जळिवो, सेठी एंटायर बॉडीटा तळे पडिजिवो. बुझि पालो कि नहीं? आउ तापर आउ गोटे जिंस होला द इंटरन्यूरॉन्स तांकर भी किछटा फंक्शन रहउची, से माने इनहिबिट भी करि परंती. जेमति तमे देखिबो जोटा कि हमें आगु को पढिबा, जेतेबेले तम सपोज योर हेड इज स्ट्राइकिंग विथ अ वॉल, मुंडटा बाडे ही जाउची गोटे कांथरे, कि गोडटा पिटी ही जाउची गोटे लुहारे. व्हाट इज द फर्स्ट थिंग यू आर डूइंग? जदि तमे जाउचो चली चली हठात तमर गोडटा जाइकी सोफा रे बाजी गला कि टेबल रो गोडरे बाजी गला, फास्ट थिंग तमे कोन करछो डॉक्टर पाकू जाउछो
तो विड्रॉ कर दो कभी
कोन करछो
तो विड्रॉल रिफ्लेक्स होती है ना तो
ओहो तमको पचार कोन करूछो को विड्रॉल रिफ्लेक्स कथा
So
छाड़ो
we
एठी
rub that place.
हां
So we rub that place or that part.
exactly you rub that place or part ସେଇଠି କ'ଣ ହଉଛି ଯେତେବେଳେ ତମେ ଗୋଟେ ଜିନିଷଟା ମାଡ଼ ହେଇଯାଉଛି ତମର ପେନ୍ ଫାଇବର୍ସ ଯେତେବେଳେ ନଉଛନ୍ତି ତମର ପେନ୍ ସେନ୍ସେସନ୍ ସେଇ ଜାଗାରେ ତମେ ତାକୁ ଟଚ୍ କରିକି ଟଚ୍ ରିଫ୍ଲେକ୍ସ ମିନ୍ସ ଟଚ୍ ତାକୁ ଦଉଛ ଟଚ୍ ସେନ୍ସେସନ୍ ସେଇ କେସ୍ରେ କ'ଣ ହଉଛି କିଛିଟା ଇଣ୍ଟରନ୍ୟୁରନ୍ସ ଆକ୍ଟିଭେଟେଡ୍ ହଉଛନ୍ତି ଯୋଉଟା କି ଏଇ ଯୋଉ ପେନ୍ ଫାଇବର ଯୋଉ ପେନ୍ ପାଥୱେଟାକୁ ଇନହିବିଟ୍ କରୁଛନ୍ତି सो कोठी गोटे जिनस माड़ हो साइमल्टेनियसली तुम जब ताको टिके रब कर दउछ कि टच कर दउछ देन योर पेन रिड्यूसेस because these interneurons they play an important role so these interneurons their presence is required because sometimes their job is to inhibit something sometimes they can stimulate something sometimes they can inhibit something बुझिएला कि नहीं
yes sir
so in case of this thing in case of monosynaptic there is no point that you can control this means तम नी जर्क रिफ्लेक्स टा हबो ही हबो तमे चाहिले भी ताको कंट्रोल कर परियोनी गेटिंग मी means there is
yes
no
sir
way तमर आंठू को जदी मु एटा पिटिली there is no way you can prevent it बट मु जदी चाहिली ताले मु जबरदस्ती गोटे माने तातिला जिंस को धरी के रखी परिबी रखी परिबो कि नहीं
yes sir
so sometimes means you can have a voluntary control over it but not in case of knee jerk reflex that is bound to happen तमे जत्ते नीटा को स्ट्रांग रखले भी तमे हेटा पिटिला माने डेफिनेटली आगो को जीवो जदी हमर वर्चुअल क्लास न ऑफलाइन ऑनलाइन क्लास न है कि ऑफलाइन क्लास है ना आई कुड हैव शोड यू गेटिंग मी सो दैट इज द केस दैट सबु जिंस सबुटी भलो ही थांदा but in case but nature has created it in such a manner that जोटा जोठी befitting से सेटा सेठी रखी so it's our job आमे सेटा केते दूर तार कोन पे सेटा रखिला आमे सेटा केते दूर देखि पारथांदे डॉग आईज रे टैपेटम लुसिडियम अछि जोटा कि शाइन करूची ह्यूमन आई रे भी थिले तो एटा आउरी बढ़िया ही थांदा dog is able to see in dark ह्यूमन आइज रे थिले तो आउरी बढ़िया ही थांदा बट किछि तो अछि there are certain limitations certain things which the nature has planned anyways any doubts on this
no sir
any doubts so in the next class there will be quiz first then we'll move to the synapse about the neurotransmitters what are the types of neurotransmitters how they are released and how they are synthesized and after getting liberated how do they function so all the things will be discussed in the next class thank you for the class you can leave now thank you
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.